Okay, good evening everyone. So we'll start with today's class. So today we are going to discuss the questions related to week 4, and today we have a set of 15 questions. We'll start. So I hope everyone knows the rules of the class, that you have to keep your audio and video mute throughout the class, and whatever you want to say, please write it down in the chat box. If you have any doubts and queries, please write down in the chat box. Okay. So yeah, today we have 15 questions. I will be showing you each question. You will be given around 3 to 4 minutes time. Think properly, and whatever you think is the correct answer, please write down your choice of correct answer in the chat box. Okay. And after that, we will discuss if the answer, what is the correct answer and what is the explanation for that answer. Okay, whoever is raising the hand, please write down your query in the chat box. Okay. Okay, and I hope I'm audible and you can see the slides, right? Yeah. So okay, let's start with the and yes, please keep noting down how many correct answers and how many wrong answers you all are giving. We will check your score at the end of the session. Okay, so we'll start with the first question for today. In a continuous stirred tank reactor with cell recycle, the relationship between hydraulic retention time and solid retention time is HRT is equal to SRT, HRT is greater than SRT, HRT is lesser than SRT, or HRT unrelated to SRT. I hope everyone is going through the lectures and joining the classes on Tuesday, right? It's very important that you go through the lecture notes and then join the class. Otherwise, you won't be able to answer the questions properly. You may not be able to understand from where I have given you this question. All the questions are from the slides, but if you have not gone through the slides, you may not be able to answer these questions. So please be disciplined in going through the lectures of that particular week before joining the live session. Okay. Okay, so your time is, if anyone has missed the previous class, they can go through the recording. I have uploaded the week 3 video in YouTube. Okay, so I hope you all are getting the links for the YouTube channel. You can go through the recording, no problem. Is everyone done? What is the correct answer to this question? It is HRT is less than SRT. So in a CSTR without recycle, HRT is equal to SRT. That is, the hydraulic retention time is equal to the solid retention time. However, with cell mass recycling, cells are retained longer in the reactor while the liquid exits. So SRT is greater than HRT. This allows for higher productivity and stability. But our question is what? In a continuous stir tank reactor with cell recycle, the relationship. So they are asking about the CSTR with cell recycle. So in that case, it will be C, where HRT is less than the SRT. Here you can see this is from this slide. So in case of CSTR with cell mass recycling, SRT is greater than HRT. Option was the opposite of this. So this is the correct answer, right? Moving on to the next question. Why are multi-stage chemostats often used for secondary metabolite production? To increase biomass yield, to separate growth and production phases, to reduce substrate inhibition, to reduce aeration costs. Start answering. Question number 2. Time starts now. Okay, let's see what is the correct answer to this question. It is B, to separate growth and production phases. So, what was the question? Why are multi-stage chemostats often used for secondary metabolite production? And the correct answer is to separate the growth and production phases. So secondary metabolites like antibiotics, we all know this term, are not growth associated. Using multi-stage chemostats allows one stage for optimal growth and another for product formation under different environmental conditions. So these multi-stage chemostats allows us to use the one's first stage for optimal growth and then the second stage for product formation. So there are two different phases. That is what they are using for secondary metabolite production under different environmental conditions. So the correct answer is D. Moving on to the next question. Which reactor type theoretically approaches the performance of a plug flow reactor as the number of tanks increases? Batch reactor, CSTR cascade, fed batch reactor, recycle reactor. So again, I am saying that um, these classes, whatever I am doing, I have to do with respect to your current year assignments given. So I am following that same pattern. I have already explained this in the last few classes, and again I'm repeating it, that I have to follow a certain pattern for this live session which I have been instructed to, and you all have to practice the questions you will be getting in your current year assignments. So same type of questions, not the exact questions actually, so that you can do your assignments properly, right? Those assignments which you are getting for every week, that carries marks, and for you to get a good score in those assignments we have been allotted, so that you can practice some similar kind of questions and have a what you can say, uh, like a revision class or a revision thing before attending your current year assignments. That's just like a practice class, so that you are well aware of what kind of questions you are getting in your current year assignments. That's all. So you all can tell me what more productive you want in this, what more extra productive you want in this, following this pattern. I'm not sure of it. I think I am trying to cover all the topics, whichever is included in the lecture notes, and you all are answering it as well, and we are also discussing the explanation for it. This is the same pattern I have been following for the last few years in the live sessions. And I am not allowing you all to talk in the class because there are many people in the class. If everyone starts talking, then I don't think I can respond to everyone. There is a limited time given for these sessions. If we keep talking, then there is no scope for any kind of productive discussion. That's why I am the only one talking, and you all are given the liberty to share your thoughts in the chat box, right? So I don't think I am doing anything wrong anywhere, but still, if you feel that this is not productive enough, you can leave the class. You can do and more productive things, right? I am not the one to take the class. I am a teaching assistant, TA, right? I am not the teacher or the professor who is going to take the class for you. This is what I clarified before also, and again I am saying it. These live sessions are a part of my teaching assistantship. So I have been given the task that you all should practice a few more questions other than the current year assignments. In current year assignments, how many questions are there? I think 10, right? Is that enough to cover all your topics from the lectures? No. That's why I am preparing a set of more questions which you all could practice. Till last year, it was that I was given questions by the Nptel team that uh, do these questions in the class. But this year, they told you prepare the questions and you discuss. So that's what I am doing. Okay. So if anyone has any problem, I think you all are being given feedback forms every week, right? I'm not sure what kind of feedback you all are giving. I It's okay. I don't want to go into that. But if you really want some different kind of live sessions, you every time you can request that we want this kind of live session and please organize for it. If everyone needs it, maybe they will start doing that too, right? Okay. So let's see question number 3 answer. It is CSTR cascade. So the question was, which reactor type theoretically approaches the performance of a plug flow reactor as the number of tanks increases? It is CSTR cascade. So as the number of CSTR in the series increases, the conversion profile becomes closer to an ideal PFR, eliminating substrate concentration gradients. So here you can see this is the slide from which this question was taken. Comparison between cascade chemostat and the plug flow reactor. Here you can see this is the large number of CSTRs. Here is the 4 CSTRs of equal size. This is the PFTR or the batch reactor, and this is the single CSTR. So it can also be shown theoretically that as the number of stages in a chemostat cascade increase, the conversion characteristics of the entire system approach those of an ideal plug flow or a mixed batch reactor. With the increase in the number of stages in the CSTR, it becomes like of a ideal plug flow or a mixed batch reactor. This is what it was written. That the question was, which reactor type theoretically approaches the performance of a plug flow reactor as the number of tanks increases. So it is CSTR cascade. Moving on to the next question. The primary function of baffles in a stirred tank fermenter is to increase temperature uniformity, to prevent vortex formation, to improve filtration efficiency, to increase oxygen solubility. Start answering. Okay, let's see what is the correct answer. It is to prevent vortex formation. So the primary function of baffles in a stir tank fermenter is to prevent vortex formation. So baffles are what? They are metal strips that break circular flow, preventing vortex formation. This enhances mixing, aeration and prevents microbial growth on vessel walls. So here you can see this is what a stirred tanked bioreactor looks like, and in this are baffles. So here you can see where is the baffle is here. This is the baffle which prevents from vortexing. Means what? Actually it is shaking. Metal strip attached radically to the wall. They are, so these this is a metal strip which is the baffle attached to the wall. They are used to prevent vortex and to improve aeration capacity. So they help in improving the aeration capacity. Baffles maintain a gap between them and the vessel wall. So this is the vessel wall, right? The orange thing. And here they are trying to maintain a gap between the vessel wall to enable scoring action, thus minimizing microbial growth on the walls or the fermenter. So they basically prevent the growth of microbes on the walls of the fermenter. That is what is the main function of baffles in a stir tanked bioreactor. Okay. Moving on to the next question. Which type of impeller is most commonly used in microbial fermentations for efficient oxygen transfer? Paddle impeller, marine propeller, Ruston turbine, flat blade impeller. Start answering. Okay, let's see what is the correct answer to this question. It is C, Ruston turbine. So the Ruston turbine provides high shear and effective gas dispersion, making it widely used in aerobic microbial cultures. So the question was which type of impeller is most commonly used in microbial fermentations for efficient oxygen transfer. So it is Ruston turbine. Here you can see what are impellers. This is how a Ruston impeller looks like. They are used for agitation, which is required to ensure that a uniform suspension of microbial cells is achieved in a homogeneous nutrient medium. So they are basically mixing so that the suspension of microbial cells is having a homogeneous mixture. It is a homogeneous nutrient medium, right? Okay. Moving on to the next question, sixth question. Which device in a fermenter is used to measure the flow rate of inlet air? Sparger, rotometer, air filter, non-return valve. Start answering. And whoever here is feeling that the classes are not productive, I think they are not going through the lectures properly. That's why I don't think they're getting the questions, and hence it is becoming like this. I know the course is difficult. I myself went through the lecture notes and it is a lot to read and understand, but there is no other option than going through the lecture notes, right? I think there are no video lectures. I think you all are only getting PPTs, right? So that is a sad part because just going through the PPTs and understanding everything is a bit difficult. I agree to that, but yeah, I think that is what is given in the course. So you have to do a little bit of more hard work and go through the lectures. If you are having any kind of doubts in the lecture notes that you could not understand this slide or that slide or this question, you can ask me here, that I can clarify. But making you all to uh, go through the class again, that is not possible here. That is not possible. Or nor I can teach you anything, that is also not possible. Yeah, I can clarify your doubts, that is possible. So rather than just complaining that the class is not productive, it will be better if you can tell me or you can ask me whatever doubts you have in the class. So for that you have to go through the lecture notes, right? Go through the lecture notes, try to understand, and whichever things you're unable to understand, that you can ask me here. How much ever time permits, I can answer you back. And if I don't know also, I can come prepared next week and answer you back, but I will answer it, right? So for that, you have to do some kind of effort first. Okay? So try to do that if you really want to make this class productive. Is it possible for the Nptel team to provide numerical explanations for the assignment after due date? I don't understand. There are numerical questions in your assignments, in your current year assignments, and you want explanation for it. Is that what you're asking? Have you checked in the lectures? Does your whatever numericals are given in the assignments, uh, is there no similar type of question in the lecture notes? I don't think so. That is the case. I will check myself. Which is the question you think is not matching with the assignment? Just let me know the question number and the week. I will check. Okay. Yeah, that will solve the thing. Okay, answer everyone. Question number six. Yeah. Okay, so everyone has answered question number six. Yeah, is it done? Week 4 assignment didn't progress yet, ma'am. What does that mean? Its due date was 16. So okay. So have you submitted the week 4 assignment? Okay. So after submitting the assignment, are you getting the correct answers? Is that it? Is it like that you get the correct answers after few days? Because I don't know the pattern of what they are doing. Week 4 due date is 20th. Yeah, I think it is not possible that your due date is coming before the live session. That is not possible. That's what I was thinking. Okay, so I think that problem is solved. Let's see what is the correct answer here. It is B, rotometer. So the question was, which device in a fermenter is used to measure the flow rate of inlet air? It is rotometer. So a rotometer measures fluid flow rate by allowing the cross-sectional area of flow to vary with pressure. So here you can see this is how a rotometer looks like. So a device that measured the flow rate of fluid in a closed tube by allowing the cross-sectional area the fluid travels through to vary, causing a measurable effect. Moving on to the next question, which type of pump is most suitable for shear sensitive biological fluids? Centrifugal pump, gear pump, peristaltic pump, propeller pump. Start answering. Hmm. Okay, let's see what is the correct answer. It is C. Peristaltic pump. So peristaltic pumps gently moves fluids by squeezing flexible tubing, preventing shear damage to the cells and proteins. So that is why it is used in the biological fluids. That is the peristaltic pump. Here you can see this is what a peristaltic pump looks like. In rotatory pumps, so that is a kind of rotatory pump, as you can see from the structure itself. In rotatory pumps, the chamber moves from inlet to discharge and back to the inlet. And a wide variety of rotatory pumps are available like gear pumps, lobe pumps, peristaltic pumps, screw pumps, camp pumps, vane pumps. And these all have relatively constant output. But the one used in the biological processes is the peristaltic pump. Okay. Moving on to the next question, which valve type is best suited for slurry and particulate matter flows? Ball valve, globe valve, diaphragm valve, gate valve. Start answering. And Shanmukpriya, I get it that you're getting confused with the options. There are a lot of formulas to use. Just tell me which question you have doubt, or which question you didn't get it, or you were unable to solve it, or you had no proper understanding of that question. I will check it. Okay, just let me know the week number and question number. Okay. And whoever is present in the class, please participate actively and try to answer the questions shown here. If you have not gone through the lecture, still try to answer it. Okay. If you give the wrong answer here also, you can have it in your memory that okay, this answer I had given wrong, this topic I could not understand, I should go back and learn it. Okay. So try to participate actively in the class. Yeah, sure, Sanmukh please do that. Chalo. Let's see what is the correct answer to this question. It is C, diaphragm valve. So diaphragm valves uses a flexible sheet that presses against a dam, making them ideal for suspension, slurries and bioprocesses fluids with solids. So the this valve, which is the diaphragm valve, that is the one which is best suited for slurry and particulate matter flows. So here you can see this is what a diaphragm valve looks like. There is an inlet and there is also an outlet. The diaphragm valves uses a flexible sheet pressed close to the edge of a solid dam to narrow the flow path of a liquid. These valves are well suited for, okay, they have kept the image here, for slurry and particulate matter flows. That's what it must be written. Okay. Okay. Moving on to the next question, the main airborne microorganisms that dominate fermentation air contaminants are gram-positive bacteria and spores, gram-negative bacteria and fungal spores, actinomycetes and protozoa, yeast and algae. Okay, start answering question number 9 it is. Okay, let's see what is the correct answer to this question. It is B, gram-negative bacteria and fungal spores. So, atmospheric air has microbial contaminants. About 50% are fungal spores and 40% are gram-negative bacteria, making filtration essential in fermentation. So, these are airborne microorganisms that can contaminate your fermentation. So, that is why it is always necessary that you have to do a filtration process in the fermentation process. First, you have to include a filtration step in the fermentation process. So, these are gram negative bacteria and fungal spores which dominate the fermentation air contaminants. Here you can see air sterilization. For an effective aerobic fermentation, the air should be completely sterile and free from all microorganisms and suspended particles. Among the microorganisms present in the air, the fungal spores 50% and gram-negative bacteria 40% dominate. Right? Okay, moving on to the next question. Which is the most common method of air sterilization in fermentation industries? UV radiation and chemicals, heat and filtration, gamma radiation, ozone treatment. Start answering. Okay, let's see what is the correct answer. It is B, heat and filtration. While multiple methods exist, industrial fermenters typically rely on filtration and sometimes heating for large scale sterilization. So, we have already discussed that one of the most common methods for air sterilization is heat and filtration. We were discussing about filtration, right? And sometimes it is heat for large scale sterilization. You can't always use filtration method for a large scale process. You have to go for heating that time. So air can be sterilized by many methods, namely filtration, heat, UV light, chemical reagents. Among these, heat and filtration are most commonly used. Okay. Moving on to the next question, the log penetration relationship for depth filtration is expressed as. So these are the formulas given. Start answering, which is the correct formula according to you. Okay, let's see what is the correct answer to this question. It is A. Okay, so the depth filtration assumes a proportional reduction per unit thickness of the filter. Integration gives this formula. So A is the correct answer. Details you can see in the slides. I have not included here. The decimal reduction time, D value in sterilization is defined as time to kill 100% of microbes, time to kill 90% of microbes at a given temperature, time to reduce population by half, time to reach thermal equilibrium. Start answering. Okay, let's see what is the correct answer. It is B. Time to kill 90% of microbes at a given temperature. This is the decimal reduction time in sterilization, where the time taken to kill 90% of the microbes. So the D value indicates how quickly microbial populations decline during heat sterilization. It is related to the thermal death constant KT. Moving on to the next question. Which statement about continuous sterilization is correct? It requires more steam than batch sterilization. It reduces nutrient loss compared to batch. It takes 5 to 6 hours for sterilization. It cannot be used in industrial fermentation. Start answering. Okay, let's see what is the correct answer. It is B. It reduces nutrient loss compared to batch. So the continuous sterilization uses high temperature, short time treatment, reducing nutrient destruction and steam consumption, that is 20 to 25% of the batch. So B is it reduces nutrient loss compared to the batch. That is why it is the correct answer. Two types of sterilization. One is batch sterilization, that is sterilizing the entire volume of medium at once using the heating, holding and cooling method. And second is the continuous sterilization, where sterilizing only a fraction of the volume at a time by using the medium as an internal heat exchanger. So what happens in batch sterilization? The entire volume of the medium is sterilized at once by using heating, holding or cooling method. Whereas in continuous sterilization, a fraction, a part of a volume at a time is sterilized using internal heat exchanger. That is what happens in continuous sterilization. Okay. Moving on to the next. Why is HTST sterilization effective in fermentation media? Microbes have higher activation energy for destruction than nutrients. Nutrients degrade faster than microbes. It avoids aeration losses. It only sterilizes gases, not liquids. Start answering. Okay, let's see what is the correct answer to this question. It is A. Microbes have higher activation energy for destruction than nutrients. Okay, why is HTST sterilization effective in fermentation media? Because microbes have higher activation energy for destruction than nutrients. Moving on to the next question. Which of the following is a non-ionizing radiation method of sterilization? Gamma rays, X rays, electron beams, ultraviolet light. Start answering. So this is the last question of today. Then we can know your scores and disperse the class. Okay, let's see what is the correct answer. It is D, UV light. So UV light damages DNA and is used for surface sterilization. Gamma X-rays and electron beams are ionizing. So the question was asked, what is the non-ionizing radiation method? So it is ultraviolet light, UV light. So here you can see radiation methods. Sterilization can be achieved using electromagnetic radiation such as electron beams, X-rays, gamma rays or irradiation by subatomic particles. Electromagnetic or particulate radiation can be energetic enough to ionize atoms. So these are ionizing radiations. Only the one which is non-ionizing radiation is the UV light irradiation, right, is useful for sterilization of surfaces and some transparent objects. UV radiation is routinely used to sterilize the interior of biological safety cabinets. So people who are have seen labs who have safety cabinets like for bacteria or for yeast for cell culture, they all have UV lights in it. Whenever we want to do some culture work, before that we always switch on the UV light for some time for sterilization so that there are no contaminants inside the cabinet, right? Yeah. So where is my mouse cursor? Okay. Yeah. So that was it for today. If you have any questions, you can write down or let us know your scores out of 50. Okay. Start typing if you want to share your score with all of us. Okay. 13 out of 15. Good. 12 out of 15. Good. 12 out of 15. Nice. Okay. So I'm telling again, if you have any kind of doubts, yeah, yeah, Ayas, you can go. You can leave the class, no problem. So, yeah, if you all have any kind of doubts in the current year assignments, then I can clarify that. But you have to tell me the question number and the week number in one class. Then I will go through it by next class and I will let you know what is, like, I can explain it properly in detail if you have not got the correct answer for it in the lecture notes. Okay, so that thing can be done. Other than that, I don't think I can teach you everything, but yeah, I can clarify some doubts whenever possible. Okay, so yeah, is there any more doubts to clear or we can disperse? No? No doubts to ask? Okay, then, thank you everyone. We'll meet again next Tuesday. Please try to be on time. Okay, thank you everyone.